All right, well, hello everybody. So seeing as the audio is working okay, it seems, and we're a couple minutes past the hour, I think we'll go ahead and get started. So welcome everybody to today's webinar. We're going to be talking about the BD Research Cloud today, so one of our newest platforms from the informatics team at BD. And as we're going to learn today, it's going to be a complete ecosystem to help support your flow cytometry studies. So I'm going to open with just one or two slides. I'm talking about what we're going to show in today's demo. And I'll also share some learning resources where you can learn more about the platform or kind of just refresh your learning after the webinar if something's not clear. So a couple of housekeeping things. First is this webinar is being recorded. So I'll show you when I get to the FlowJo website at the end of the slides where you'll be able to find this recording once it's posted in a few days, as well as the rest of our learning resources. In addition, if you have questions at any point, please feel free to put them in the chat. You don't have the ability to unmute, but you can type them in the chat and I'll do my best to keep an eye on it throughout the presentation and I'll try to answer them in real time. Otherwise, you can post them at the end. There'll be time for questions, or feel free to send me an email. Here's my contact information. It's just my first name and my last name at bd.com. So feel free to send any inquiries you have about the platform after today's presentation. And with that being said, I think I'll go ahead and get started. So for those of you who've never seen me on these webinars before, my name is Veronica Obregon-Perko, and I'm an application scientist with BD. And so most of the time I spend my day talking about FlowJo, our flow analysis software. But as I mentioned, we're going to be talking about the BD Research Cloud today. So again, a platform for supporting your flow cytometry studies. So what exactly can this platform do for us, right? So this is just an overview of all of the different functionality that can be found within the BD Research Cloud. And so even though cloud would imply that it's just a file storage solution, I can assure you it does a lot more than that, and it's kind of summarized here on this slide. And so really our goal here with this platform was to try to address some of the sticky points that arise when we're working with flow cytometry studies. How do we stay organized? Where do we put all of our files? How can we design panels in an efficient way, revisit them, reorder reagents from them, and also maybe plan ahead for experiments that we're going to be doing at the cytometer. And so we've tried to encompass all of that on this platform for you to try to facilitate some of those sticky points that we often run into or we wish there were simpler solutions for. So really at the heart of the BD Research Cloud is the cytometer, right? The cytometer is going to dictate what kind of panels we can do, what our experiments look like, because you can only put certain colors, right, on certain filters and certain lasers. So there's no point in, exploring reagents that are not compatible with your cytometer. So really at the heart of a lot of this functionality is going to be the ability to either upload or create cytometer configurations within the BD Research Cloud. And so there you can store information like what kind of lasers are on that cytometer, as well as what kind of filters or detectors, and what kinds of parameters, or I should say what colors, fluorophores, you can use on each of those different filters, right? So really facilitating the downstream processes we're going to talk about. Lastly, there's a reagent resource where you can browse the BD catalog, but you can also manage an inventory. So if you have a lot of antibodies in the lab and you want to keep track of them all to help with reordering, help with panel design, we've incorporated some functionality for that as well. But you can also just explore the BD catalog within the platform during panel building or just during looking at the reagents alone themselves. And you can look at their spectral spillover and see if it's going to be a good candidate for your experiments or for your panel. And in terms of workflows, right, that feeds directly from the cytometers and from the reagents, right? So when we design panels, or for anyone who's ever designed a panel, you know that you typically have 5 different sheets of paper in front of you, right? You have your cytometer configuration, so you know which colors are going to be available to you. You probably have the catalog open for some kind of vendor of choice so that you know what color combinations are available for your antigen of interest. And you're going to have an antigen density chart, probably, maybe some kind of gating strategy. And so it's a lot of information to look at once. But what we've tried to do is, based on the 2 features before this, incorporate all this into a single interface so that when you build your panels, you can see reagents that are available, but also reagents compatible with your cytometer. Okay, so these all kind of feed and interconnect with one another. And in addition to making panels, you can even make procedures like protocols that then you can share with people on the platform or make them interactive if you have offsite collaborators. And also one of my favorite features is the ability to make BD Diva templates away from the cytometer. So for those of you that are familiar with BD instruments for cytometry, you know that Diva is typically the acquisition software. And sometimes we're short on time at the cytometer and we don't have time to label each of our tubes the way we want to, or label each of our colors because we're running out of time at the machine. And so wouldn't it be great if there was an ability to pre-label all of that before you got to the cytometer? So we've provided you with an opportunity to do that. And again, this is going to be able to bring in labels from panels that you've designed based on your cytometer configuration. Okay, so it's all going to be based off cytometer configurations that you're using pre-existing or that you've uploaded to match what's available in your lab or available in your core. And so after you make these templates, right, you can bring them right into Diva, acquire your samples. And once you've acquired them, where can you store all that information? Well, as the name would imply, in the Research Cloud, there is a file storage capability as well. So there's an area to store your FCS files. And even better, it has a direct link with FlowJo Software. So if you're going to use FlowJo for your analysis, then you'll be able to open your files from the Research Cloud directly within FlowJo. So you don't have to play the download, upload game, no installation of external apps or anything like that on your computer. So it's really just facilitating that process of obtaining your data faster and in a more efficient way. Okay, so these are some of the features we're going to talk about today in today's demo. And I think that's pretty much it for slides. I really just want to spend the majority of the time in the platform itself. So let's go ahead and take a field trip to the FlowJo website. So I can first show you a couple of resources, and then we'll get started with the demo. So if you go over to flowjo.com, head on over to Learn, and head over to Webinars. This is where you'll see not only our upcoming webinars, including the one you're watching right now, but if you scroll all the way down and hit this Recorded Webinars button, that will bring you to our library of previously recorded webinars, including BD Research Cloud. I think we've only had one before this one, but the one from today will be posted right around here. They're typically in chronological order. So today's recording will be located here if you want to retrieve it or share it with one of your lab mates. Now, you can also learn a lot about the platform. So let's say you've watched the webinar, but you're still kind of iffy on something that you learned, you want to rewatch it, right? If you head over to the Learn tab and go over to FlowJo University, these are going to be short videos, kind of just reminding you how to execute some of the functionality that we've gone over in today's webinar, right? So you can see they're organized by product. We're talking about the Research Cloud today. And if you hit Start here, right, you'll see that there's two different cards, right? How to get your lab organized and start putting in some data. And you can see there's a couple of short tutorials here. They're typically less than 5 minutes. And so you have an opportunity to rewatch them without committing to a whole 1 hour webinar. Okay. So a lot of learning resources to be explored here. So don't become overwhelmed with all the information. You can always revisit it at a later time. Oh, question in the chat so far. Can you also create FACS chorus experiments offline in addition to FACS Diva? Richard, that's a great question. So we cannot at the moment, but I think that's a great suggestion. And I know it's been a topic of discussion within our team to address that acquisition software as well. But at the moment, we can only do FACS Diva. All right, so having looked at these learning resources, okay, and if you still want to learn more about the platform, we have an entire web page dedicated to that product where you have an opportunity to, ask for quotes, learn about the different licensing options and what they can do for you. You can see that these accounts start free. Okay, so right after today's webinar, you can go make an account and start, playing around with the tool and seeing what it can do for you in your lab. And you can see that you get access to a lot of our features without paying anything. Now, if you start running out of storage, you want to unlock some additional functionality, then you'll have an opportunity to upgrade if you would like to. But the individual and essentials license are free. So I definitely recommend starting there, inviting some of your lab mates, your friends, and see what it can do for you and send us your feedback. All right, so moving on, let's go ahead and get started. So to go to the Research Cloud, it's all on the internet. It's not like it's an app that you have to download or a software you have to install. Okay, it's just bdresearchcloud.com. Right? If you go to the landing page and you don't have an account at this time, you can hit create account and you'll have an opportunity to do that. Okay, but assuming I have an account already, which would be tied to your FlowJo portal if you have one of those already, then you can log in, right, with your same password. All right, and then it's going to bring you to your landing page, right? So now we're in the BD Research Cloud. Now, when you do first sign up, you'll automatically be given what's called an individual organization, which means it's just you alone in there. So it's your own personal space to, create panels, upload files, anything you'd like to do in there. So you'll automatically be given that organization. But with an essentials license, you can create an organization, right? And you can see that I've been invited to a bunch of them here. You can create your own and invite users, right? So if, let's say I have created my own and it's called My Lab. If I go over to people, users, here I have an opportunity to invite different people to my organization so that we can collaborate together. Okay? And you can also create groups and projects, right? So you can segregate your members of your organization into different groups. You can also create projects within groups. Okay, so there's a lot of nested hierarchy here to keep you organized, or you can keep it nice and simple and just have one group and one project that you all work on together. Okay? Or you can make it more complex if you're working on some kind of a, like a major clinical trial, for instance, where there's typically teams of individuals dedicated to different aspects of the study, right? You can separate that all right here. Right, so let's go through some of these resources one at a time, right, and kind of explain what they can do for us. So the first one I want to point out is cytometers. And the reason for that is because, as I stressed in the slides, this is really going to be the heart of the functionality here. The cytometer configuration is ultimately going to determine what kind of panels we can build and what kind of experiments we can make. Okay? So what you can do in your organization is you can upload cytometers of your own, right? So how can you do that? Well, you can hit the create button. And there's two options for bringing in a custom cytometer. One is you can import a BD Facts Diva file, which will have the configuration for you. Okay, so you see there's a button here to upload that configuration file. And again, this comes off BD instruments that are running Facts Diva. Now, if you don't have a configuration file, let's say you're working with a different cytometer or a cytometer that runs Chorus maybe, you can just build a custom cytometer instead. Okay, so the way you can do that is head over to create a custom cytometer. Here you can give it a name, vendor, model, right? All of these different informations about the cytometer itself, even its location. And then you start adding really the meat and potatoes of the cytometer, which is the lasers and the filters, right? So you can begin by adding one laser, putting the wavelength. You can put what kind of detectors are off that laser, okay? And if you have multiple lasers, right, you can add additional lasers and the corresponding detectors for each of those lasers. Okay? So you can navigate through this interface, and ultimately what it'll look like is something like this. So here's a custom cytometer that was uploaded by a collaborator in this organization. And this is what it looked like when she was done. Okay, she put the configuration, which is a 5 laser, and it's showing the breakdown of how many for each color. Or how many filters for each color laser, right? And you can see grouped by laser here, and we've got the laser properties there. We've also got the filters, and you can even take it a step further. And within each filter, you can go ahead and list out some parameters or some fluorophores that are compatible with each of those filters, right? So this is information that is, going to be here for you to reference when you're looking to build your panel. But as we'll learn in a bit, this platform is going to call to this information automatically for you and only present you with options that are compatible with the cytometer based on the filters and based on the properties of that fluorochrome. Okay? So a lot of value in uploading a cytometer that matches the configuration in your lab or in your core if you use a core facility. Now, let's say you don't have access to those resources, or you're just in a pinch, you want to play around with the resources that are available to you now. We've gone ahead and uploaded some of our most popular cytometers as well. So if you go to the BD Cytometers tab, you can see that our whole fleet of cytometers are already preloaded for you. So you can start designing panels if one of these configurations matches very closely to what you have in your lab. Okay, and if you want to look at the configuration, you just have to select the cytometer and you'll be presented with that information. And you can even export it as a PDF. So if you want to print it out, keep it on your desk or take it with you to the core, you can have that information for you as well. All right, so that's the heart of the cytometers there. So having uploaded a cytometer or knowing that we have these BD ones available, it's really going to simplify that panel design process for us. Okay, so how does that work here? Well, let's head over to research and we're going to go to workflows and panels. And what we're going to do here is create a panel, right? Let's walk through that panel design process and talk about how that cytometer configuration kind of fits in. So we're going to hit this drop down, make sure it says create panel, and then we're going to hit create. Now at this point, I'll be prompted to name it. So, let's make this a rather straightforward panel. Let's call it CD4, CD8 panel. Oh, not CD9, CD8. So CD4, CD8 panel, right? We're going to assign it to a project, right? You can see in this organization, I've got several projects here to keep us organized. So let's just go ahead and assign it to my COVID study. If I've built panels before, I can kind of bring in those markers so I don't have to start from scratch. But this is a brand new panel, so let's pretend I don't have anything to go off of. So I'll go ahead and hit start the process. Okay. First step, select a cytometer, right? So why do we want to do this? Again, I've been stressing this a lot, but just to, beat a dead horse, it's really just to say that is choosing this cytometer configuration is ultimately going to help us in the subsequent steps, because then the Research Cloud will only present me with fluorochrome options that are compatible with that cytometer, right? I'm not going to be interested in designing a panel with colors that I can't even use on my instrument because I don't have the right laser or I don't have the right filter, right? So why not just eliminate those possibilities by going ahead and designating a cytometer from the start? Okay? Now, if I have any custom cytometers, they'll show up here. Otherwise, I can just ask to see the BD ones that have been uploaded automatically for me. Right, and I have an opportunity to pick any of those cytometers. So I'm going to go ahead and just pick the Symphony A5. So BD Symphony A5, it's even reminding me what the configuration looks like, and it's giving me an opportunity to download it one more time in case I want it for my lab notebook or something. Okay. So I've selected my cytometer, right, which is going to be the Symphony A5. We're going to complete that step. All right, next step is going to be, well, what are you trying to measure, right? What are the antigens that are going to be in your panel? Okay, so there's two ways you can bring antigens into this panel. One is to utilize kind of our population resource. So first, let's pick a species, let's say human. And this is a CD4, CD8 panel, so I definitely am interested in CD4 T cells. So you can see that I have an opportunity to add just the CD4 antigen, or I can add CD4 T cells as a population. And what that's going to do is preload antigens that are kind of minimally required to identify that population based off the literature. Okay, so if I pick that population and add it, you can see I have a population of CD4 T cells, and it's given me CD3, CD4, CD19, and CD45. Now, I don't have to take it all at face value. If there's some of these that I find redundant or I don't want, I can always remove it if I'd like to using the little trash icon. Now, let's say there's individual antigens I want to add, right? I can still do an individual antigen search. So maybe in this case, I want CD8 alpha. So I can look at CD8 T cells, and I'll add that antigen as well. I can also add populations that I'm kind of curating here myself, really just for my own information, right? So if I hit this plus button, I can create a new population, right? And I can even call that CD8 T cells. And then again, at this point, I could definitely just proceed, right? Everything at this point is kind of optional. But if you do want to give yourself some information to help you with the panel design process on the subsequent steps, then you can go ahead and input that now. So for instance, CD4 T cells, we know are probably going to be high for CD3, right? And we know they're going to probably be high for CD4, and they're probably going to be negative for CD8, right? And so the reason this information could be helpful to you in the panel design process is because, long story short, there's rules to follow when designing a nice panel, right? So we want to put bright colors on dimmer antigens and dimmer colors on brighter antigens. So if I know that information about the antigen up front, I can go ahead and just curate it here. And that way on the subsequent step, it'll help me when I start pairing those fluorophores. Okay, so we're not going to talk a whole lot about principles of panel design. That's a topic for another webinar. But we've tried to keep those in mind, right? Keep those rules in mind for designing a good panel and give you the tools to input that here. Okay. And so I could do the same thing here if I wanted. There's also an opportunity to designate a clone. Okay, so if you know that you have an absolute favorite CD4 clone and you're not interested in seeing what's available in these other clones, you can go ahead and dictate that now. I only want to see colors available in L200 for anti-CD4. Or you can leave it open and it'll just show you everything we've got. Okay. You can also do a dump channel, right? That's what this exclusion here is. So granted, I've got a lot of colors here to play with. I'm not really going to run out of room. But if I had a really low parameter cytometer, having dump channels can be a way to kind of leave you some room in the panel, right? So that you can put in other antigens, right? So for instance, if I know that my CD4 and CD8 T cells are going to be negative for CD14 and CD19, I might as well put CD14 and 19 off the same fluorophore, save myself some room. So you can definitely create dump channels as well. You can also include a viability dye, always highly recommended, but you know, some folks choose not to use it. But if you would like to include a viability dye in your panel, just hit this toggle and it's going to present you with some of our options for viability dyes as well, right off of our catalog. So I'm going to go ahead and delete CD14 and CD19 for now. Now, the other reason these populations might come in handy is to watch for co-expression, right? So let's say I have two activation markers that I'm interested in, like CD38, right, and CD69, for instance. And I know I'm going to be looking for CD4 T cells that are double positive, right, for that marker. You can go ahead and, you know, dictate that on your individual populations, or I can make another population here and call it activated CD4 T cells. And just kind of be cognizant of the fact that I'm going to need to be able to resolve CD4 cells that are single positive for CD38, single positive for CD69, or a combination of both, right? And so using that information, right, you'll want to avoid spillover spread between those antigens. So again, another way to utilize the populations resource here. All right, so I'm going to go ahead and get rid of those as well. Let's just keep this panel simple for today's demo. So again, select your species. You can add antigens either individually or as a population. You can then assign antigen densities to help you with the fluorochrome pairing process on the next step. You can designate a clone if you'd like. You can also create dump channels. Right, so let's move on, go to the next step. Right, now this is the interface that's really going to help out with the panel design process. Right, so again, for anyone who's ever designed a panel, it's a lot to think about at once. Right, you have to first think about the density of your antigens, right, how bright they are, how dim they are, how abundant they are on certain populations. You have to think about spillover between colors, right? We want to try to spread out colors as much as we can across detectors. And going into the antigen density, we also want to consider the brightness of a color, right? We don't want to put a really bright color on CD3 because it's just really going to saturate that system. So we want to reserve the brightest colors for the dimmest antigens and vice versa. So what we've tried to do here is put all that information in one view, right? So why have 10 Excel sheets open when you can just have a single page to really guide you through all that information? Okay, so how do we start the antigen pairing process? Well, I can sort this definitely if I want to. So for here, I can sort by antigen name, or I can sort by antigen density or fluorochrome availability, right? So if you want to pair the ones where there's less colors available first, you can do that, right? So let's say I do that. And so there's less colors available for the viability dye. So maybe I want to do that first. I select the viability dye, and you can see that that list has changed. It's presenting me with everything that's available for viability dye from the BD catalog. Okay, so we're not going to present you with options that you can't even buy. We're only going to show you what's available in our catalog. And again, compatible for your cytometer based on the cytometer you picked earlier in the process. So there's my viability dye, right? I've got some choices here. I can sort them by laser. I can even sort them by brightness if I'd like to. Okay, so if I want maybe the dimmest ones first. So actually, I'm kind of liking this 520 option here, right? I can see where it peaks in my cytometer configuration here. And if I'm happy with that color choice, I can hit pair, and you can see it snaps to the top of the screen. I can unpair it if I change my mind. Now moving on, I'm ready to do CD3, and you can see that FITSI and AmCyan have kind of gotten this red message on there. This is basically warning me that these colors are going to peak in the same filter, right? So FITSI is 488 blue, and I already am using 488 blue off the 51520 filter for my viability dye. Okay? Now, the reason we give you this warning is because, you know, it's going to be hard to compensate those two colors, if not impossible, on a traditional instrument. But if you do have a spectral instrument that's able to unmix these through spectral unmixing, then you could pick it if you wanted to. Okay, but usually in a traditional cytometer, even in a spectral cytometer, you want to try to pick colors that are very different from one another because they'll be much easier to resolve. Okay, so we give you that warning, but you can override it if you still want it. Okay. Now, if you don't want to get confused, you don't want to see those, go ahead and just hit this button that says hide occupied filters, get those out of the way. You can even hide color choices that are already taken. Okay. All right, so knowing that, let's see, I'm going to maybe go with something a little dimmer. Maybe I want to pick a different laser. Let's go with Alexa Fluor 700. All right, I can interrogate spillover between these two, which there's virtually none. Okay, but I can peek at that here. CD4, right? Maybe I want to pick another color, maybe go something violet, right? I can pick that color, right? If I'm worried about spillover, I can go ahead and preview that again. I have the opportunity to do that. Okay, so I'm going to pick CD4. I'll go ahead and pair it to this violet detector here. And then same for CD8, and maybe I'll pick something else, maybe something off the UV. Okay. And so you can see I've paired all of my antigens, so they've snapped at the top here. I can take one last peek and make sure I'm not dealing with any major spillover issues. Okay, so spillover into another detector, you're going to see that with this dashed line, right? Whereas the maximum emission, you're going to see with the solid histogram. Okay. So once I'm satisfied with my panel, right, I can complete that step. And I have an opportunity to start designating what I might want to order. Okay, so I have a suggestion here for Brilliant Stain Buffer. Here's everything I chose during the panel design process, but I can also look at different sizes, different clones, right? Select all of those, complete that step, review it one more time, maybe trash things I don't want to buy. Like, oh, you know, I don't need Brilliant Stain Buffer. I just got some. Let me just go ahead and get rid of that. And I'll go ahead and finalize this workflow. And my panel is now designed. But I'll have an opportunity to ask for a quote or download the shopping list if I'd like to. Right, so let me try and open that again. Here we go. Now it's nice and loaded. So I can go ahead and add them directly to the BD cart. So if your lab has a BD account and this is how you do your ordering, right, you can see that everything I designed is showing up here. So I don't have to go search for it after I design my panel. It's already done it for me. Or I can email it or download it, give it to my lab manager, or order it through some kind of punch out system at my university. You can also ask for a quote. All right, so that's the panel design process in a nutshell. So once you've designed your panel, where can you see it? Well, if you go back to your workflows dashboard, go over to the completed tab, here it is, CD4, CD8 panel, right? If I open up that workflow, the first thing it'll show me are the steps that I did to make that panel, but really what I want to see are the pairings, right? So if I go to the workflow details, head over to that third step where we first picked those fluorochromes, right, you can kind of see that, revisit that information again, right? Here's what I paired to CD8 to CD3. There's all the properties of the fluorochromes, as well as what filter and laser they're assigned to on my cytometer configuration. Okay? So you can revisit panels you've designed, print out summaries for them as well. Now let's talk about some of the other resources available and kind of how they feed into the reagent selector, right? So we noticed two things in the reagent selector. One is that those populations you're defining, you can definitely define them in that interface, right? Remember when we were adding antigens, we had an opportunity to create populations, but that's coming from a resource within the Research Cloud. The other thing that it's pulling is reagents from the BD catalog, right? So where is all that information coming from? So let's talk about the populations first. So if we go over to resources, populations, right? Here's all of the curated ones we've already provided for you, right? And if you click on a population, you can see kind of, again, minimum antigens that might be required to detect that population based on the literature. But what if you have, a super awesome cell type that you want to add at this stage, right? So if I look for one created by my collaborator here, you can see that she's added a few of her own, right? She has a Treg population that she measures on a regular basis, right? And she's gone ahead and added that information on how to locate that cell type, right? So not a gating strategy per se, but kind of an antigen profile for that population. Now in doing this, when you're designing your panel on that step where we added populations, if you type that population name into the search, it's going to automatically load these antigens as well as their properties, okay? So that's where those populations are coming from this curated resource in the populations area. And you can also create a custom population, right? So you can see there's even a prompt to do it up here, create population. You can name it, give the species, add all the individual markers. You can even put links to publications in case you want to come back and revisit something. Now, the other resource is the reagents, right? So again, that's all being pulled from the BD catalog, which you can browse here in this interface. Right? You can see we've got 19,000 different options for our antibody fluorophore pairings. And so this is what's being called to when you're designing your panel. But it's filtered by what's available for your cytometer and what's available for purchase for that antigen. Right? Now, let's say you have reagents in your lab that don't come from BD, right? That's a very real possibility. You can go over to the custom reagents tab and make your own reagent, right? So you can see that I have colleagues that have already made some custom ones. Let's say we have a tetramer stain that we had, built for us, right? If you go ahead and hit that antigen reagent resource, right, you can see that there's some properties here for it, right? It's an APC tetramer. And then you can see there's some other ones here, like 7AAD viability, FIT-C for a different antigen. But the benefit of adding these custom reagents, one is that you can add them to your inventory, which we'll talk about inventories in a second. But then you can use these reagents when you're building your panel. Okay, so when you're presented with all the fluorochrome options for a particular antigen, you'll be shown this color option because BDRC knows, or the Research Cloud knows, that you have this available to use in your lab. So if I'm trying to find colors to pair to my tetramer antigen, right, it's going to show me APC because it knows I have that available. Okay? So you can go ahead and customize this experience as well as the populations resource. Within reagents, there's also, again, an opportunity filter I'm trying to get it to go into, or that it's compatible with my cytometer of choice. Okay. And then lastly, there's an inventory function. Okay, you can import inventories in bulk through a CSV file. You can also, if you ordered something off the BD catalog, you can hit the add to inventory button and it'll show up in your inventory. And you get a lot of information in the inventory, right? As much as you want to put, including lot numbers. even where it's located down to the box, right? Box 1, row 3, column 7, right? You can put all that information. You can even add notes about, if you've noticed something weird about that antibody, or you want to remember that somebody gave it to you, so that, you can hit them up for it again if you run out. Okay, so a lot of information here that you can put for your inventory. Okay. Now I'll add that the inventory is one of our paid features. Okay, so if you sign up for a free account through Individual or Essentials, you won't have access to this. But if you do upgrade, you'll be able to manage an inventory for your reagents. Okay. But all the other 3 tabs will be available to you. All right, so we talked about, let's see, cytometers, building a panel, kind of where those panel resources come from, which is going to be in the form of the BD catalog and custom reagents, as well as custom populations. So let's talk about some of the other resources available in the Research Cloud. One is one of my favorites, which is the experiment builder. So if we head over to resources experiments, right, we talked about the pain point of sometimes working at the cytometer is that you don't have time to label everything the way you want it to. So how can we maybe do that away from the cytometer? Well, we've tried to create an interface here that looks very familiar to Diva, as we'll see. And it gives you an opportunity to kind of knock all that out before you get to the cytometer. Okay, so you can see I've got some saved here. Let's create a new one. Okay, so if I'm going to create a new experiment, and let's give it a name. So, given that we just designed a CD4, CD8 panel, maybe I just want to call this CD8, CD4 tubes. Again, put it to a project. Now we're going to pick a cytometer. Why do we want to do that? Well, 2 reasons. One is because then we'll only be presented with the filter options for that cytometer, right? I'm not going to label filters that I'm not going to use in my experiment because they don't exist on that cytometer. And also, as we're going to see in a second, we'll be able to link it to a panel to inherit those labels. Okay? So you'll pick your cytometer, right? It's going to remind you of the configuration. And this currently is only compatible for BD cytometers running FACS-Diva. So you'll just need to pick your FACS-Diva version. And when I hit that experiment, you'll notice that this is going to look a lot like Diva for those of you that have used it. Okay, so I have an opportunity here to label specimens and tubes and even add them. Okay, so if I have a specimen, let's say these are blood samples, and I've got one animal, right, or let's say one donor. And then I can add Diva, you have to kind of sit here and, painstakingly type your labels. And yeah, once you've typed all of them, you can kind of copy and paste. But we've integrated an ability to just bring in your labels right from the panel you've designed. Okay, so if I select everything here, go over to select panel. I'm going to look for my CD4, CD8 panel, select that. I'm going to say apply panel. And we can hide unused parameters, right? So filters you're not using, right? Let's just get rid of those. And here we go. There's that panel that I just designed earlier, and it's got all the labels I want, the antigen and the color, right? I can even add the clone if I want to right here. So another way to think about this functionality is, let's say you don't need to design any panels, right? All your panels in your lab have already been designed. Then go ahead and just upload it anyway to the panel design process, right? Pick all those colors that you already use, and that way you can make use of this functionality very quickly, right? So imagine being able to make Diva experiments, in a matter of minutes here, just by, applying labels to panels that we're already actively using in the lab. Now, again, this is minimal information here. You want to take it a step further. You can also add, actually, let's go ahead and save it first. Let's go ahead and add keywords, right? So keywords are metadata that are associated with your FCS files. For those of you that use FlowJo a lot, you can add keywords in FlowJo, but they don't become inherent to the file itself. It's really just kind of a display in FlowJo, right? So good practice is to, maybe annotate as much as you'd like to on the actual file. And that way there's never any question of what that file is or where it came from. Right? So let's say, for instance, I have 2 different donors here, and I want to designate, right, a time point for my study. and add that keyword column and say, oh, well, both of these were actually from day 1. And it's not like these are going to show up in the name of the file, but they'll show up as metadata when you inspect the files in a software like R or FlowJo. Right, I can have another one for something like, for let's say group. Right, I can add that, and maybe one of my donors is a control, right, and maybe my other donor is vaccinated. So you can add as much information as you'd like to, right, to annotate these specimens and tubes. Lastly, taking it one step further, right, you can go over to plots. Right, so you're not going to be able to draw gates on these plots, but if you want to start making your global worksheet, right, maybe I'm going to start with some scatter gates here. So I've gone ahead and added those plots, right? I'm going to have a forward scatter plot for singlets, another forward scatter plot for debris, right? But I can also add, something else, like if I have a viability die, right? Add that histogram plot, go ahead and leave it there as a placeholder. All right, and that's it. So I've got my experiment. Right now I have an opportunity to export it. Okay, so it's going to come as a zipped file. Now, how are you going to bring that into Diva? Well, I can show you quickly, right? So I've got a Diva offline station here. Now, bear with me, it's not connected to a cytometer because I don't have a cytometer in my home, unfortunately. But I can still run the software just to show you how you would bring this file in. Okay, so you would have a folder for under your user login. And once you hit that folder, you can go to File, Import, Experiment, right? And we're going to bring in that template we just made, CD4, CD8 tubes, import that in, right? If I launch that experiment, we'll just ignore that message. It's just telling me I don't have a cytometer, which I know. Then I can go ahead and see everything I created, right? Here's my blood specimen, donor 1, donor 2. If I select donor 1, I can go to labels, and I'll see those labels from my panel have come through. And if I go to keywords, right, I can see the keywords that I associated with it, including time point, which was day 1, and group, which was the control, right? And same for the second donor as well. If I head over to the global worksheets, right, look at that global sheet 1, here's those plots that I added, right? Scatter properties, as well as viability die. Now, I will say this one doesn't have any labels because I'm not connected to a cytometer, but if you were, it would match the labels that you see on the samples, right? So here it says viability die 520, it would say that here on the x-axis. So you can see how, much easier that can be, especially if you're trying to get to the cytometer to build this experiment, but you're running short on time, or the next user is behind you ready to get on. So you can try to build this as much as possible on your computer. And that way, when you get to the cytometer, all you have to do is maybe add your compensation controls and draw some gates. All right, so let's go back to the Research Cloud and let's exit out of that experiment. Now, before I leave this resource, I'll tell you that you can also build an experiment as a plate editor. Okay, so if I create another experiment here, all right, and let's just call this CD4, CD8. Plate, okay, just so I can show you that functionality quickly. We'll pick the same cytometer, right, everything the same, but this time I'll start with a plate editor view, just so I can show you that. Okay, so then you can go ahead and pick your plate size, right, and then here you have an ability to kind of dictate what your plate's going to look like, right? So maybe my first row is always my compensation controls. I can add that or leave them empty and add those in the Diva software. I can also add, other specimens here. Okay. Right, so I can add all of those labels. And then if I go to the following page, right, which is where we typically apply our labels, right, I'm going to go ahead and apply these quickly so I can show you. So here's my CD4, CD8 panel, apply those labels. Right, and here I can actually download a PDF. I think, one sticky point with plates is that we sometimes lose our place on what's on the plate, right? Or we forget how we laid it out. Well, you can make a report for that, right? So here's my plate. It's reminding me, what each of those specimens were, which panel I applied. So if you have multiple panels on the same plate, right, that can get kind of confusing. So you can be able to reference that panel down here. Looks like it's having some trouble loading. I might have made it a little too big. But in any case, here you can see that you're able to get a nice layout for each of the plate experiments that you've made. All right, so I'll go ahead and exit. I'll save that and then go ahead and exit out of this. All right, so the last two things I'll show you is the File Explorer and then workflows, and then we can see if there's any questions. So let's say you run your files at the cytometer, right? So now you've generated these FCS files. Well, where can you store them? Well, if you go to the Research Cloud, you can upload them to our File Explorer. Okay? So you can see here we've organized some folders based on the different projects, right? So if I have my COVID projects here, there's also a titration experiment that's been done. So here I can go ahead and upload files using the upload button. And you can upload files that aren't FCS or workspace, right? So if you did have PDFs or Excel sheets you wanted to bring in here, you certainly can do that. But you can see here we've got a number of different FCS files and workspaces that have been created. Now you can email them to yourself and download them to, give them to a collaborator or something. But if you're downloading them just for the sake of analysis, there is an integration feature in FlowJo to facilitate that process. Okay, so let's open up FlowJo real quick. And so here within FlowJo, you can have an opportunity to say, let me go back to the FlowJo tab. So I can add samples from BD Research Cloud, right? So I can have a direct link to the BD Research Cloud. I don't have to download the files first and then drag them in. I can come here, navigate to the organization where I have the files that I want to retrieve, go to the project that they're stored in. Here we go. I've got maybe this FCS file that I want to analyze. I'm going to go ahead and bring that in. Okay. So here's my FCS file. Okay, so now I've done some analysis. Oh, I drew 1 or 2 gates. All right, called that cells. Okay. And so this is an analysis I want to save, right? So let's just say it's more complex. Right now I just drew 1 gate, but I think you get the idea. And so I'm ready to save this. Well, again, I don't have to save it and then upload it back to the Research Cloud. I can do all that from this interface. So I can say File, Save As, BD Research Cloud Workspace, right? It's going to open up a similar dialog window, basically asking me, well, where do you want to save it? Which project do you want to put it under? And then I'll have an ability to retrieve it again. Okay, so here I'm going to go ahead and save it in this location. I'll keep it with just today's date. I'll go ahead and hit save, right? And it's even reminding me that I'm working from the cloud with this file. Okay. Now, let's say I come back another day. I'm ready to retrieve this workspace. How can I do that? All right, let's go ahead and save those changes. And let's go ahead and quit FlowJo, pretend we're starting a new session. Right, and if I go back to the Research Cloud and go back to my individual org, right, I'll likely be able to see that in my File Explorer now. There it is, December 14th, right? This is the workspace we just made. Okay. So I go out of that. Let's go back to FlowJo quickly. All right, and now this time I'm going to say open, but I'm going to open from the Research Cloud. Again, so I didn't have to download it. I can just open it directly within the software here, navigate to where that folder was. There it is. There's that December 14th workspace. I'm going to go ahead and open it. Depending how large the file is, it might take a minute or 2 to open, but you shouldn't have any lagging during the analysis. It might just take a second to open the first time. And so you'll see it'll open and it'll retain that FCS file, so I don't have to reconnect FCS files, and that same analysis will still be there as well. Okay. While I'm waiting for this to import here, I'll pause and see if there's any questions in the chat. If there are any questions, feel free to post them at this time if you'd like to. I can always revisit the chat. And here it is, right? There's that FCS file, the analysis of a single gate that I drew. Okay, but you can see again, it's all calling from the Research Cloud. So it's all interconnected. Okay, so last feature I'll show with the remaining minutes we have here. I'll go over back to the workflow section, which is where we built our panel. But you can also create, actually, I'm going to go back to my collaboration organization. You can also create workflows that are kind of like protocols, right? So after you've created one, like what can it look like? Well, let's open one that was made by my collaborator here, where she had a few different procedures, but within each procedure, there's steps, right? So kind of like sub-steps. So she had a procedure for thawing and counting cells, right? And she gave details for each of those steps. And she even assigned it to one of my lab mates, like, oh, this needs to be completed by Dan. And she left a note saying that she was running low on media. Okay. Right, and so you can execute each of these steps, right? You can see that most of them have been done, right? There's one step we're waiting on, which is acquiring our cells. That's why it's active. And once my collaborator, Emily, starts that step, right, she can mark it as complete. So you can see the step is active. She can leave notes if she'd like to. And once she finalizes this workflow, then it'll be complete. And she can revisit this protocol in the workflows section. Okay. So how do you go about making something like that? Well, instead of create panel, it'll be create custom workflow. You hit the create button, right? And you'll be asked to give it a title, right? So if I'm making a protocol for a surface stain, I'm going to assign it to a project, right? Maybe require that my lab mates put notes on there, move forward. And then the interface will look something like this, right? You can have a procedure. Within that procedure, right, you can have different steps. You can rename the procedure. Okay, so if I go add a procedure here, right? This is going to be my surface stain. I can say create. Okay, and now I can add different steps, right? So maybe my first step is I do a live dead stain of some kind. Right, I can save that, right? I'm going to delete that procedure if I don't need it. Okay. And you can see that's the first step, and I can keep going, adding multiple steps, putting as much detail as I'd like to. I can assign it to different group members. And then once it's done, you just publish the study, and published studies will basically end up in the active section so they can be executed. If you haven't finished it, you want to come back and edit. We have some that are in design, right? Including the one I just tried to make right now. And then you'll have completed workflows that you can revisit, right? If you want to see notes for what was completed in that workflow. And you can also turn it into a study. What does that mean? A study is basically a workflow that repeats. Okay? So if you have a workflow that you do every morning, like maybe calibrating your PCR machine or calibrating your small cytometer, you can have this reset every morning, right? You can see there's a little symbol here. This is a recurring workflow. So every morning this resets so that me and my collaborators can let each other know that we completed this process. Okay? So that's what a study is, basically a recurring workflow. Okay. All right, and I think that is all I wanted to show in today's demo. I know it's kind of a whirlwind of everything available in the cloud. I think moving forward, we're going to have webinars that are a little more focused on individual features, but we definitely just want to give a rundown of what's available right now for people. So again, accounts start free, so you can definitely go and make an account for an individual org. You can also, see if I can find that information for you. Here we go. You can upgrade immediately to an essentials license and invite 5 people to work with, or I'm sorry, up to 10. You can invite up to 10 people to work with. And you guys can start seeing if this is a tool that's going to help you in your laboratory. And if you feel like you want to unlock more, you want more storage, more features, you can talk to us about upgrading to a professional or a premium account. Okay, but just know that these start free. So it's definitely worth trying out. Let's see, there's something in the chat here. There is a question about if the BD Cloud complies with 21 CFR. That's a good question. So does it directly? No. And I think that's true for a lot of software out there. Out of the box, it's not 21 CFR compliant. Now, that being said, there can probably be ways to use it in a 21 CFR compliant manner. So you can reach out to me and I can try to help you figure all those details out. But we are aware that that is a highly sought after feature, for clinical studies as well as for contract research organizations. So we are working to be, cognizant of that and how we can make that the platform more friendly for it. But there's likely ways to meet some of the requirements that might be required for your study in particular. So if you're interested, feel free to reach out to me and we can try to work out how one would use this platform in a 21 CFR compliant manner, but that doesn't mean it's necessarily going to meet every single requirement. That's a good question. All right. I guess I'll see, are there any other questions? Does anybody have any questions about any of the tools, about where to get an account? Go ahead and leave my contact info up here one last time. All right, well, I'll hang around for another minute or two. If there's any questions, feel free to put them in the chat. Otherwise, for the rest of you, I'll bid you a good rest of your day and a happy holidays. And I hope that you all have an enjoyable rest of the year and come back nice and refreshed for next year. And you continue to join us for our webinar sessions. And of course, as I've mentioned multiple times, feel free to reach out if you have any questions at any point, and feel free to visit our resources if anything is still unclear, so you can kind of enhance your learning. So thank you all for those who joined today.